So our last video was really largely about the history of this whole idea of evolution by natural selection. At the end, I did start to talk a little bit about kind of the mechanics behind the process of natural selection, but I left you with a bit of a cliffhanger, which is this question about how can we actually tell whether a population is evolving, whether that evolution occurs through the process of natural selection or through other means. And to start exploring this question, I'm going to redefine evolution in a slightly different way, which is as a change in allele frequencies in a population over time. So that starts to raise a question of what is a population exactly? And I'm not going to completely answer that question right now, but I'm going to kind of hint at that a little bit. And I'll use squirrels as an example for the time being. So this is a picture taken from a paper all about coloration in one of our North American squirrel species. It's one that doesn't quite make it this far into New York, but this is fox squirrels. And fox squirrels vary in color. And so the coloration for coat color for fox squirrels is genetically determined. And so the biologists that conducted this study went out and surveyed a bunch of squirrel populations and characterized coat color and genetics in those different populations. And so a given squirrel population in this case would be a bunch of squirrels in one of the local regions that's illustrated on the map here. And apologies, this is a little bit gruesome, but biology is a full circle of life. So this is a photo of kind of the variation in squirrel coat color that was found as part of this study. Okay, so anyway, so talking about evolution by natural selection. So I wrapped up that last lecture video kind of running through this process. The idea is that if you have a population that has some kind of phenotypic variation with a genetic basis to it, so there are genes and alleles that code for a particular trait, and within that population, biological fitness varies. So depending on what genotype and phenotype you have, there are different outcomes in terms of your survival and ability to reproduce. And then, so what that means is then versions of individuals that have greater biological fitness wind up producing more offspring. The net result of these conditions and these processes is a change in allele frequencies in a population over time. So that's the process of evolution by natural selection in a nutshell. And it kind of helps to see a concrete example of this. So here's a little cartoon from a website on evolution. So this is just showing a beetle population where beetles vary in color. And you can imagine many bird predators are very visual. So they use sight to seek out tasty things. And so it might be that there are birds that prey on these beetles that have learned how to find the green beetles really easily. So they preferentially eat green beetles. And what that means is these orange beetles are not being eaten at the same frequency as the green beetles. And so over a series of generations, what may wind up happening is replacement of the green beetles in the population with orange beetles instead. But we have to step back for a moment to the 1900s to a dilemma. At the time, the question was, okay, well, we know that there's something going on with regards to evolution by natural selection, but how can we actually tell if a population is actively experiencing this? And for thinking about this, we have to be aware that Darwin didn't know anything about Mendel's work. So remember that Mendel died in complete obscurity. And so Darwin never knew how the traits that he wrote about were passed on from one generation to the next. He had some ideas, but nothing solid. So it took the rediscovery of Mendel's work in the early 1900s to then lead to motivation to try and connect the dots between what we now knew about how traits get inherited and this process of natural selection that Darwin had identified. Okay, so biologists have this puzzle, and I think this part's kind of funny. There are a lot of debates over how to resolve this situation, and there's a British mathematician who happened to learn about all of these heated debates, who kind of looked at the biologists and said, you know, the math actually isn't that hard for figuring this out. So this is G.W. Hardy pictured here, and this picture is just perfect for that. You know, why are you making a big fuss about this, biologists? So he figured out a method for answering this question, and actually it was worked out at about the same time by another guy who wasn't necessarily who you might think would make such a pivotal contribution. W. Weinberg, who was a German obstetrician gynecologist and came up with the exact same idea. So we actually give both of these guys credit for this concept. So for working through this, we're going to consider the simplest possible case to start out with, kind of like how we started with Mendelian genetics. So we're going to think about a situation where an individual's phenotype is determined by a single gene that gets inherited in a dominant recessive fashion. And for answering this question, we're actually going to start out by thinking about kind of a null prediction. So what do you expect if a population is not experiencing natural selection? So a population that's constant through time. And we're going to ask the question of, if we know something about the frequency of alleles in that population, then what can we come up with in terms of the genotypic and phenotypic ratios that we'd expect to observe if there isn't any natural selection? And we're going to refer to that null condition as the Hardy-Weinberg equilibrium condition. So just to kind of reiterate that here, so we're thinking about a case where we have a single gene and two alleles, dominant and recessive. And so if we were to look at a population and look at individuals in that population, and you know, we can use maybe red for the big A allele, and how about blue for the little a allele, right? So we might have some individuals that are homozygous dominant in our population, some individuals that are homozygous recessive in our population, and then some individuals that are heterozygous. It's going to be a little tricky to draw here, but I'll do my best. So those are heterozygotes. Okay, so here's our question. So there are 5 assumptions associated with a population being in this equilibrium condition. And these are assumptions that you will need to know. And I'm going to give you a little mnemonic device for remembering them, which is large, random, M, and M's. Okay, so here's what they are. So the assumptions are, there aren't any mutations, so we're not going to have an allele B suddenly show up. Next assumption is random mating. So this is an assumption that every gamete that gets produced by each of these individuals in the population has an equal chance of bumping into every other gamete. Third assumption, so here again, this is to help us determine when natural selection is happening, is there's no natural selection present. So there's no association between genotype here and an individual's biological fitness. Fourth assumption is that this is an extremely large population size. What I've illustrated here is actually not that big. You need to be up at least a couple hundred to have a sufficiently large population size. And then the last assumption is that there is no migration. So that means there aren't any individuals entering this population or leaving this population. So what that means is they're not taking these alleles with them. There's no gene flow in or out of the population. So again, so just for this mnemonic, so we have a large population size, random mating. We have no migration, no mutations, and no natural selection at work for those assumptions. Okay, so what we're going to do is we're going to take some ideas from thinking about individual crosses to population level calculations. So when we think about a cross between two heterozygous individuals, so we're thinking about big A crossed with big A little a, each of these individuals, the first step in that cross is thinking about what kinds of gametes do they make. And they each make a pool of gametes that essentially pair up randomly. And so if we cross these two, there's an equal number of alleles in this cross, right? Each parent has a big A allele, so there's two big A alleles, and then each parent also has a little A allele, so there's two little A alleles. So if we're thinking about the percentage of offspring of each genotype, hopefully you can think about how many of the big A, little A, or little A offspring you'd expect, right? That's just our plain old simple Punnett square, right? So, parent number one is going to produce 50% big A, 50% little a gametes. Parent number two is 50%, 50%. So we're going to wind up with offspring with 25% big A, 50% big A, and 25% little a. And the thing I want you to realize now is that we can apply the same type of calculation to figure out genotype frequencies in a population. So in the current population here, there's an equal number of big A and little a alleles. And Harding-Weinberg kind of added a little bit of math to this. And so instead of just referring to big A's and little a's, they defined P as equal to the frequency of the big A allele, right? So in this case, the frequency of that allele is 50% is going to be 0.5. And then they designated Q to be the frequency of the other allele. So in this case, the little a allele, and so that's 0.5, right? And then we need to think about kind of random mixing of these alleles in a population. So here's kind of how they defined what your expectations are for the frequency of homozygous dominant individuals, heterozygous individuals, and homozygous recessive individuals you'd expect in the population. So this is simply kind of taking that Punnett square and throwing P and Q on it, and then multiplying those things together to see what we wind up getting. So I'm just going to rewrite this Punnett square here so we can think about it a little more. Okay, so we have big A and little a, right? And if big A is is equivalent to P, and 50% of the big A alleles in the population or 50% of the alleles in the population are the big A allele. So this is P, and that's equal to 0.5. And then the other 50% are then the little a allele, so that's Q, also equal to 0.5. So then if we wanted to figure out the genotype frequencies in the population, this is going to be P times P, or P squared, equal to that. Each of these is going to be PQ, and then we're also going to wind up with Q squared. So one other principle that's also going to be true is if we add together the frequencies of these different genotypes, the total will equal to 1. In other words, all of them will be accounted for. So what that means is there are two kind of mathematical relationships here, right? So if we're dealing with a population where there are two different alleles, then if you add the frequency of each of those alleles together, that should add up to 1, right? So P and Q together should always be equal to 1. So it's going to be equal to all the alleles in the population. And similarly, if we add up all the genotype frequencies in the population, that's P squared plus 2PQ plus Q squared, classic quadratic expansion, that will also be equal to 1, or all of them. Okay, so fairly simple when you have equal allele frequencies for P and Q, 0.5 each. But there are cases in populations where there are unequal numbers of alleles. And just to give you one example, talked quite a while ago about how even though the O blood type allele, so that's recessive, it's actually the most common allele in the human population. And similar things will be the case for things like the allele that causes sickle cell disease, or the sex-linked allele that causes color blindness. Those allele frequencies they're not equal in the population. There are more people that have one allele type than the other allele type. So that's a situation where we're thinking now about P, or the frequency of one allele, as being a lot bigger, like say 0.9, and the frequency of the other would then be 0.1 to add up to all of them, right? So now looking at this, we need to think about, okay, well, what would we expect for the genotype frequencies in the population? And to get there, what we need to do is apply the same mathematical process and calculate that out. So what we're picturing is, in this population, 90% of the alleles are the big A, 10% are little a. And what we want to do is just kind of pull the individuals apart. So we're thinking about just their alleles, and then combine them back together to see what the genotype frequencies are. So in this case, so P is going to be equal to 0.9. Q is going to be equal to 0.1. So if we want to figure out our genotype frequencies, right, big A is going to be equal to P squared. So it's going to be 0.9 squared. So that's 0.81. Okay? Big A little a is going to be equal to 2PQ, right? So that's 2PQ, 2 times 0.9 times 0.1. So that's going to be 0.18. And then we can do the same thing for Q squared to figure that out. Or we can remember that we have to account for all the genotypes in the population. So for little a, that's going to be equal to Q squared. So that's 0.1 squared, 0.01. So 1% of the population. So when 90% of the alleles in the population as a whole are the dominant allele, notice that the majority of the recessive alleles, so 19% of the population has at least one recessive allele, the majority of those recessive alleles are in the heterozygotes. So it's homozygous recessive individuals are very rare when we have these kinds of allele frequencies. Okay, right, so we can use this expansion, the P squared plus 2PQ plus Q squared equals 1, to go from our allele frequencies to our genotype frequencies, right? But you can actually also calculate back to the allele frequencies from the genotype frequencies if you know your population is in Hardy-Weinberg equilibrium. So let's take a look at that. So let's say we're now thinking about, one of my favorite human genetic traits, tongue rolling. And tongue rolling is a dominant trait in humans. So if we were to symbolize this, we might put a capital R for tongue rolling. So let's say we survey a human population and we find out that 9% of people surveyed, so what this means is lowercase r is going to be unable to roll the tongue. 9% of the population cannot roll their tongue. So that's little r. So that's equal to a frequency, population frequency of 0.09, right? So now if we assume this trait is in Hardy-Weinberg equilibrium, can we calculate the frequency of the recessive allele in the population? So here we are given, if we're thinking about P's and Q's, we've just been given Q squared and told that Q squared equals 0.09. And if we need to calculate the frequency of the recessive allele in the population, this is asking us to find Q. So all we need to do is take the square root of Q squared, the square root of 0.09, and so we'll find as Q is equal to 3. Okay, so what that means is, so there's only 9% of people that can't roll their tongues, but 30% of the alleles in the population are the little r allele. So the next question here is, now what proportion of people would you expect to be big R, and big R, little r? So now we need to find big R, and big R, little r. Okay, so here's where we're going to start. So we now know Q. We know this is a population where there's only 2 possibilities. So Q is our frequency of the little r allele. So if we want to figure out the frequency of the big R allele in the population, that's going to be P. And that's going to be equal to 1 minus Q, because P and Q have to add up to 1. So that means that P is 0.7 in this population. And if we want to figure out big R, and big R, little r, then we just need to expand back out. So big R is going to be equal to P squared. So now that we know P, that's 0.7 squared, 0.49. And then for big R, little r, right, that's going to be equal to 2PQ. So 2 times 0.7 times 0.3. So that's 0.42. So then we know, 49% of the population is homozygous dominant, able to roll their tongues, and 42% of the population is heterozygous. Okay, so in this case, it's crucial to realize 2 things. So first, that for this question, the question said we can assume Hardy-Weinberg equilibrium. And then the other thing is just to realize that, how to identify, your P's and your Q's. So remembering that P and Q, the allele frequencies, have to add up to 1, and that then you can also use the expansion to figure out the percentage of, or frequency for the homozygous dominant or heterozygous individuals. Okay, so in some ways the Hardy-Weinberg equilibrium itself is a little bit boring because it shows what the null expectations are for a stable population. So this is a population where from generation to generation the allele frequencies are remaining constant. And so what that means is that we can always calculate the genotypic frequencies from the allele frequencies. That's what we just did right here, right? But in real life, the question we're actually interested in is what could make this change? What would make this change is a violation of one of those assumptions of the Hardy-Weinberg equilibrium, right? So there's those 5 assumptions, right? And a violation of any of those assumptions indicates that a population has changed or that it is changing genetically. So we don't have stable allele frequencies over time anymore. So let's just take a look at those assumptions again and at what happens if a given assumption is violated. And we're going to talk about these in more detail in a little while. So first off, what about the assumption of no mutations? So what we have to think about for this is mutations are something that happens to individuals. So if all of a sudden there are mutations in the population, maybe one allele out of 1,000 alleles in a population is now changed. That's not really going to have a major impact on allele frequency across the population. So even if the mutation assumption is violated, generally no major impact. How about the assumption of random mating? The opposite of this is something called assortative mating. So that would be something where for this tongue rolling example, for example, maybe the individuals who can roll their tongues don't reproduce with the individuals that can't roll their tongues. So they only mate with other individuals that are similar to them. If that happens, that can actually cause a real change in the genotype frequencies in a population, right? Moving away from this expectation here. So third assumption, what if there is natural selection? So that's going to result in change in allele frequencies and genotype frequencies across generations. And so that is the process of evolution taking place. There's also that assumption of an extremely large population size. And the contrast to that is a small population. And we'll look in more detail at what that does. But it basically means that allele frequencies in the population will start to fluctuate over time. And there's a couple of different terms associated with that happening. One of those genetic drift, and the other is a specific circumstance called the bottleneck effect. And then the last assumption again was no migration or no gene flow. So none of these alleles moving out of the population or back in. And if there is migration, and migration of a small population size, sometimes you can get something that's called the founder effect. So basically, anytime any of these assumptions for the Hardy-Weinberg equilibrium doesn't hold, or they're violated by something, that means there's going to be some really interesting stuff potentially happening in that population. So I'm going to conclude this video with an example of use of the Hardy-Weinberg equilibrium, because oftentimes this feels really abstract. Kind of when do people actually use this? And this comes from some work conducted by a couple of my research mentors and advisors. So they're studying migration in Africanized honeybees. So a bit of background on this. When European settlers arrived in the US, they brought European honeybees with them. So the honeybees that we think of that make honey that we like to eat, and the honeybees that pollinate a lot of commercial crops, those are actually migrants. They're European honeybees. And whenever people bring animals to a new place, some of them escape. So the European honeybees did exactly that, and they established wild populations in the US. They actually didn't do so well in South America. And so South Americans, were pretty interested in also having access to honey. So in the 1960s, what they did was they started trying to introduce different types of bees into South America to try and figure out a variety of bee that might do better there. So the African honeybee was introduced in Brazil to try and interbreed it with European honeybees to produce a gentle bee that could also handle being in a tropical climate. And unfortunately, the bees escaped once again and started new colonies in the wild and started to spread throughout the neotropics. Also, unfortunately, the bees that escaped happened to be fairly aggressive, so very counter to the original goals of this breeding project. And by 1993, people were starting to find some of these Africanized bees at the US border. So here's a map of the southern states of the US showing the spread of Africanized honeybees by year across counties. So again, in the late 90s, early 2000s, bees started showing up across the southern US. And then as time has gone on, they've been found further and further to the north. The good news is they have not moved any further north than what you see here, because it turns out they're not very good at surviving cold winters. But there's a lot of interesting stuff happening in that time frame in the southern US. So my research mentors had a question. Now that's a place where these Africanized honeybees are going to start encountering some of the feral European honeybees as well. So, how might that interaction go? There are kind of two major possibilities. One is that the Africanized honeybees, could be so aggressive that they outcompete the European honeybees and replace them. That's one possibility. Another possibility is that the Africanized honeybees might wind up hybridizing, so interbreeding, with European honeybees. And that does assume that hybrid bees are biologically fit, so able to survive and reproduce. So they set about testing these two hypotheses by genotyping the bees in two different ways. So one thing they did is they genotyped a mitochondrial gene to tell whether the queen bee was Africanized or European. I have the question here of why. The reason why is so mitochondria are always inherited from the mother. So if the mother was Africanized, she would have an Africanized bee mitochondrial gene. So they genotyped the mitochondria, and then they checked allele frequencies for another gene against the expectations for Hardy-Weinberg equilibrium. And the marker they used for the second round of genotyping is something called an allozyme, which is basically a protein enzyme that has different functional forms. So when you genotype using an allozyme, it's very similar to just directly genotyping an individual. And you kind of get something that looks similar to the outcome of gel electrophoresis as well. So you get different protein forms based on different alleles, and in this case they genotyped for a metabolic protein called malate dehydrogenase. And so here's a gel that shows different genotypes for malate dehydrogenase, and there's two main alleles. There's a fast allele and a slow allele. So here's individuals that have the fast genotype, so two fast alleles. Here's an individual that has the slow allele, and then here's some examples of individuals that are heterozygous, one fast, one slow. Okay. So they went out and they collected bees right after they're first noticed in the southern US. And so here is genotyping of the bees in 1994 when they were all European. And so you can see the distribution of the fast, slow, and slow genotypes. And then here in 1995 is where the Africanized bees first arrived. And what I hope you see in this picture is that the genotypic frequencies between the European and African honeybees are different, right? So there's a greater representation of the slow allele in the European honeybees and a greater representation of the fast allele in the Africanized honeybees. And so keep in mind there are those two hypotheses. One hypothesis was that it could be that the Africanized bees are going to outcompete the European bees. And if that is the case, what we would predict is that over time, we should see these Africanized honeybee allele frequencies stay the same as they are when the bees first arrived. However, if that's not the case, and if instead what happens is Africanized and European honeybees hybridize with each other, then what we'd expect to observe is a change from Hardy-Weinberg equilibrium over time. So some disruption, and we'd expect to start seeing new allele frequencies in both of the bee types, Africanized and European, and then eventually a return to Hardy-Weinberg equilibrium. So just to reiterate that, if Africanized bees outcompete the European ones, we expect these allele frequencies to stay constant through time. But if these two bee varieties interbreed with each other, they have very different allele frequencies to start out with. So we'd expect a deviation from Hardy-Weinberg equilibrium and then eventually a return to that. So here is what was found. So immediately after the Africanized bees arrived, what they found was for the first couple of years, the bee varieties were not in Hardy-Weinberg equilibrium. But by 1997, what I hope you can see here is the allele frequencies have started to merge between the two bee strains, and they actually went back into Hardy-Weinberg equilibrium after that. So my research mentors concluded, okay, that provides support for hypothesis number 2. What happened when they arrived was the Africanized honeybees hybridized with the European honeybees. So I have a question here for you to think about, which is, which out of those Hardy-Weinberg assumptions got violated? And I'm going to leave that question for you to think about and come back to later. And we can ask the same type of question in other biological contexts too. So there's variation in squirrel coat color. If we genotype individuals and examine allele frequencies and compare them against what we'd expect for Hardy-Weinberg equilibrium, that could tell us whether squirrel coat color is experiencing natural selection and evolving through time.